Hello, my name is Dr. Roderick L. Roll, and today I will be talking about antimicrobial methods for controlling microorganisms. Drugs are chemicals that affect physiology in any manner. It can be a positive effect or a negative effect. A positive effect is when a drug is going to cure some disease. A negative effect is like an opiate that can cause an analgesic effect that makes an individual a habitual drug user. We are also going to talk about chemotherapeutic agents. These are drugs that act against disease. You most commonly know about the drugs that affect cancer, but chemotherapeutic drugs are any drug that will negatively affect the disease. One of those chemotherapeutic agents that we will discuss are antibiotics. So antimicrobial agents are drugs that treat bacterial infections. These are going to be called antibiotics. So they're going to negatively affect the bacteria, the bacterial infection. In this slide, we're looking at a bacterial lawn. This was done with a hockey stick where bacteria was smeared throughout this petri dish. Then a chemical was soaked into a disc. Then the disc was dropped directly on the bacterial lawn, and notice how the chemical is inhibiting the growth right around the disc. Therefore, a halo will appear. If you look at all of those discs, one of them will produce a larger halo than the other. That chemical that's in the disc is an antibiotic. Some bacteria produce antibiotics. How you discover antibiotics is when you're growing bacteria, if you see a halo around a certain colony, then that bacteria, that colony, needs to be further studied. The gram-positive filamentous streptomycetes belongs to a family of bacteria called actinomycetes. About half of the antibiotics and semi-synthetic antibiotics are produced from streptomyces. In this slide, we're looking at different examples of bacteria that produce antibiotics. Again, the family actinomycetes are gram-positive, filamentous with branches. Majority of these are streptomycetes. So the most common bacteria that will produce the antibiotics will belong to this category. Semi-synthetic antibiotics have a slightly altered chemical structure from the original. When you alter a natural occurring antibiotic, you make it more efficient. You make it last longer in the presence of very harsh chemicals that the bacteria can produce to try to deactivate it. In this picture, we're looking at some drugs that have been chemically altered. Once it is chemically altered, it has a better efficacy. The original drug discovery in the 1930s were a sulfur-containing drug called sulfonamide. All sulfur drugs were variations of p-aminobenzene sulfonamide. In this talk, we're going to discuss several ways to stop bacterial growth. We're going to discuss an inhibition of cell wall synthesis, inhibition of protein synthesis, disruption of cytoplasmic membrane, inhibition of general metabolic pathways, and inhibition of DNA and RNA synthesis. The first one we will discuss is inhibition of metabolic pathways. The metabolic pathway involved in DNA synthesis of bacteria will require a molecule called PABA. This stands for para-aminobenzoic acid. This molecule must be present in order for the bacteria to make DNA. The first step that must occur is enzyme 1 must convert PABA into dihydrofolic acid. If this enzyme converts its reactant to a product, then enzyme 2 must take dihydroxyfolic acid and convert it into the next product. This sequential event will ultimately produce DNA. Sulfa drugs are metabolic analogs of PABA. So look in the picture at the structure of PABA. Now look at the other chemical compounds in this picture and notice that they all have the same basic structure. So therefore they are analogs for PABA. These analogs for PABA have the ability to compete for the same active site that PABA competes for. Therefore, if the sulfur drug binds, it will inhibit the production of the dihydrofolic acid. In the picture, the analog would be the inhibitor in image A. The substrate would be PABA. Here is the biochemical pathways to make DNA synthesis. PABA will be converted into folic acid, which is a vitamin. Folic acid will be converted into dihydrofolic acid. Dihydrofolic acid will be converted into tetrahydrofolic acid. Tetrahydrofolic acid will be converted into folinic acid. And then folinic acid will be used to make DNA. If you interfere with any of these steps, DNA will not be created. Through competitive inhibition, a PABA analog will interrupt dihydrofolic acid production. This will ultimately stop DNA synthesis. In the picture below, where it says C, you see that the PABA analog is going to bind into the active site of the enzyme. PABA will not. Therefore, you will not produce dihydrofolic acid. Will this affect humans? No. Taking a sulfonamide or a sulfur drug will not affect humans in a negative way because humans consume folic acid, which is one of those steps. Bacteria do not consume vitamin B, but we do. And these are the foods that it is found in. Newer sulfur drugs like Bactrim are used. These drugs are better because they are combination drugs. It is composed of two different drugs, Sulfamethoxazole and a Trimethoprim. When you combine these two drugs, you have better killing capability in that Bactrim drug. Sulfamethoxazole is going to stop the synthesis of dihydrofolic acid. That is indicated by the red star. The Trimethoprim is going to stop the synthesis of tetrahydrofolic acid. That is indicated by the green star. So this sulfur drug, Bactrim, is a combination of drugs that will ultimately still stop DNA synthesis. It is just more efficient. Other synthetic agents like sulfones are related to sulfonamides. These sulfones will also interfere with folic acid production, therefore stopping DNA synthesis. The example is dapsone. That drug, if you look at it, has a sulfur containing unit. This drug is used to treat Mycobacterium, which is the active bacteria that causes tuberculosis and leprosy. Now we're going to move on and talk about drugs that affect the cell wall. We're going to first discuss the structure of the cell wall. Then once you understand the structure, then we're going to look at trying to destroy it or trying to inhibit the production of. What does the cell wall of bacteria look like? In this image, we're looking at a gram-positive bacteria and a gram-negative. What we are interested in right now is the thickness of the peptidoglycan. Notice that in the gram-positive bacteria, you have about 40 layers of peptidoglycan layers. In the bacteria to the right, gram-negative, you only have 1 to 5 layers of peptidoglycans. What is the peptidoglycan? It is composed of two major things. Peptido is referring to the tetrapeptide, and glycan is referring to the sugar. So collectively, they make up the peptidoglycan. Let's talk about the sugar and the glycan. This is nothing more than a modified glucose molecule. So N-acetylglucosamine is also called NAG, and N-acetylmuramic acid is also called NAM. Look at the structure of those two molecules and notice how similar they are to glucose. The only difference is carbon 2 and carbon 3. They have modifications there. Each NAG and NAM layer of the bacterial cell wall is held to the next layer by a pentapeptide. This pentapeptide is made of glycine. So they're going to call it a pentaglycine. Or it can be called a connecting chain or a cross-link. Notice how it is perpendicular to the layers. The layers are labeled in this image 1, 2, 3, 4, and 5. So each layer is composed of NAG-NAM, these repetitive sugars, or modified sugars, and they make up these rows, these layers. So in this image, we are also looking at the layers. They're also numbered 1, 2, and 3. What holds layer 1 to layer 2 is the pentapeptide or pentaglycine. This is a series of 5 glycine molecules which are amino acids linked together covalently. Also in this image, we have a tetrapeptide. The tetrapeptide is going to be attached to the NAM. And in the tetrapeptide, you have L-alanine, D-glutamine, L-lysine, and D-alanine. They are also covalently linked. How do drugs affect the cell wall? Beta-lactams are a category of drug that have functional groups that contain a beta-lactam ring. So in this image, I have an arrow going to the beta-lactam ring so you can see the structure of it. So any drug that has that same ring would fall under the category of beta-lactams. In this image, I have listed several examples of beta-lactams. We have penicillins, we have cephalosporin, and we have monobactrim. So all 3 of these are categories of beta-lactams. Notice how they all have the same beta-lactam ring. Natural penicillin and other natural beta-lactams will affect gram-positive bacteria and spirochetes. Gram-negative bacteria contain an outer membrane. This will not allow these drugs to access the cell wall. So look in the picture, and I've highlighted the outer membrane. So remember, the gram-negative has a smaller peptidoglycan, but outside of that, it has a membrane that can restrict molecules from coming across. If it restricts the molecule, then the beta-lactam cannot destroy the cell wall. But if another drug was to destroy the outer membrane, then the beta-lactam could get to the actual small peptidoglycan in gram-negative bacteria. Or if penicillin was allowed to come through a porin, a hole, a doorway, then penicillin, which is a beta-lactam, could destroy the cell wall. Since that is rare to happen, we have chemically altered these beta-lactams. By chemically altering the beta-lactam, we've made it have a broader killing range. So now it can kill gram-positive and gram-negative. But natural beta-lactams have a much narrower range of killing. It might only kill gram-positive. How is synthesis of the wall actually inhibited? So in this image, what we're looking at is 2 main sugar units, the NAM versus the NAG. The NAM has these cross links attached. This is how layer 1 is attached to layer 2, layer 3, and layer 4. That cross link is a pentapeptide that is made of glycine. If you can somehow block the production of that pentapeptide, if you can somehow remove that purple block, then layer 1 will not be attached to layer 2 and layer 3 versus layer 4. The beta-lactams that we just discussed will inhibit linkage between NAM and NAM. The beta-lactams will stop the production of that purple block that's in the picture. So if you look at the picture, we now have a green block. So that means no pentapeptide will be formed. In that case, the peptidoglycan layer will form, but it will not be anchored to the pre-existing layer. If you cannot attach the new layers to the older layers, then the bacteria will become very weakened in reference to their cell wall. If the cell wall is weakened and becomes porous, then water can rush in or water can rush out. The bacteria will eventually lyse. In order for a bacteria to produce, it must insert about 2 million new subunits. So if the NAM and NAM are not linking together, then you are producing the layer, but not technically producing it to stick to the bacteria. Therefore, the bacteria is not adding 2 million new subunits. In this image, we're looking at a bacteria that is not exposed to beta-lactam and one that is exposed to beta-lactam. The beta-lactam makes the bacterial wall weakened. Therefore, the bacteria is beginning to lyse. The next drug category is called vancomycin. Vancomycin is going to be a drug that will bind to the terminus of the tetrapeptide. The last amino acid that is on the tetrapeptide is called D-alanine. If I somehow block the D-alanine from binding to the pentaglycine molecule, then the layer 1 will not bind to layer 2, and layer 2 will not bind to layer 3. But you will still produce the layers, but they will not be able to attach to one another. So that is what this drug category will do. Vancomycin is a drug that will bind to the D-alanine and block its interaction. Another category of drug is called cycloserine. Cycloserine competitively inhibits 2 cytoplasmic enzymes that are in the bacteria. One of those enzymes is going to convert L-alanine to D-alanine. In the picture below, I have that conversion. So cycloserine is going to inhibit enzyme 1 from doing its job. So if we cannot produce D-alanine, then you do not have a complete tetrapeptide. And remember, the tetrapeptide is a cross bridge that binds to NAM. The second enzyme is going to incorporate the D-alanine into the tetrapeptide. So cycloserine is going to block that process also. Therefore, I have a little purple shield. So if I block the incorporation of the D-alanine into the tetrapeptide, I therefore also will block the layers from forming. The next drug category we will talk about is bacitracin. Bacitracin is a highly toxic drug that must be applied topically. This drug is going to prevent the secretion of the actual NAG and NAM subunits. The last category of drugs we will talk about will affect bacteria that produce tuberculosis and leprosy. That bacteria is Mycobacterium. Mycobacterium is a very, very difficult bacteria to inhibit or destroy because it has a very thick, waxy mycolic acid layer. So this bacteria is a gram-positive bacteria, and it has this outer coat. This outer coat is made of mycolic acid. So these two drugs, asanazide and ethambutol, will both destroy this mycobacterium by disrupting the formation of the mycolic acid layer. Since this bacteria reproduces every 12 to 24 hours, it will take the drug months to even years to treat. How are some bacteria resistant to this process? So some of the drugs that we give patients are somehow deactivated. So let's talk about those. Beta-lactams can be deactivated by penicillinase. So penicillinase is an enzyme that will split the beta-lactam ring, therefore making the beta-lactam inactive. When treating penicillin-resistant Staphylococcus infection, you must use a semisynthetic penicillin called methicillin. In the image below, you see the difference in the natural penicillin and the chemically modified version called methicillin. If Staphylococcus is resistant to methicillin, then you have to use something like vancomycin. Resistant Staphylococcus to methicillin is actually called MRSA, methicillin-resistant Staph aureus. In that case, vancomycin is going to block the interaction of the tetrapeptide to the pentapeptide. This is going to stop the formation of the layers that make up the wall.